Thank you so much for coming, especially since we really weren't sure this morning if anyone would make it through, so I really appreciate it. And I hope you all enjoy the festive dinner that we have today. It was a little treat from the caterers. I'm very pleased to be able to introduce Dr. Martin Alda. He's a professor and Killam Chair in mood disorders in the Department of Psychiatry at Dalhousie University. He received his medical degree from Charles University in Prague, and he trained in psychiatry at Charles University and the University of Ottawa. Prior to coming to Dalhousie, he held the Canada Research Chair in Bipolar Mood Disorder at McGill, and he has published over 160 original papers and book chapters and serves on editorial boards of several journals. Today, Dr. Alda's talk will focus on the pharmacogenetics of bipolar disorder and some of the results of studies that characterize the phenotype of lithium response using linkage, association, and sequencing strategies. So please welcome Dr. Alda.
Good afternoon. Can you hear me well? Is that okay? Okay. Thank you, Sue, for the kind invitation and the introduction. And when I was thinking about what to present, I was not exactly sure how to balance some of the clinical and phenotypic aspects and some of the molecular work that we've been involved with. So it will be a mix of those two, and please feel free to interrupt with any questions you may have as we speak, and let's start. So, for those of you who are not psychiatrists, I would like to start with just a little case vignette that would give you some sense of what we are looking at in the area of mood disorders. This is a lady that we saw 9 years ago for a family study of bipolar disorder, and she was 50 years then. And her history was this: when she was 16, she had an episode of depression that lasted for a few weeks. She was prescribed some medication that she couldn't remember. She took it for a few months and then she stopped and was fine. Then in her 20s, she had a number of episodes that would last typically about a week when she would be busy, quite happy with racing thoughts. She would be speeded up, she would speak fast, and busy around the house doing things. And these episodes would usually take a few days to a week and then would subside. Then when she was 28, she had an episode of postpartum depression that lasted for about 2 months. And at that time, she was quite typically depressed. She had low mood, she was tearful, she felt guilty for no apparent reason, she felt hopeless, was not interested in anything, she was tired, she couldn't sleep at the same time, she couldn't concentrate, and then again the episode subsided. And then over the next several years, she had every month about 2 to 3 days of clearly decreased mood, and similar to that particular episode, just lasting a few days. Then when she was 31, she had another episode, and it was similar to the previous one. She went to see a psychiatrist at that time, and in the meantime, she also became pregnant, and suddenly the episode went away, and she went through the pregnancy. After this third pregnancy, third delivery, she developed another postpartum depression, and at that time, she saw the psychiatrist again, and the psychiatrist started her on lithium, which is prophylactic treatment to prevent the episodes of depression and mania from coming back. And then for over 20 years, she was stable on lithium. She had just one minor breakthrough episode that didn't require any intervention, but for 20 years she was completely well and functioned as if she had no illness whatsoever. But then in 2006, so this was a couple of years after we saw her, her family physician noticed that her creatinine was increased, indicating some possible compromise of kidney function. And so the family physician decided to take her off the lithium treatment because there is a possibility that some of the impairment could be ascribed to the lithium treatment. After she stopped the lithium, she was fine for a period of time, but then she developed more symptoms, and so her doctor started her on another medication, topiramate, which is an anticonvulsant, first 50 and then 100 milligrams. But she was not that well, and her relatives noticed that she was maybe more withdrawn, more quiet, and then in 2009 she killed herself. And this is not an uncommon story. What this brings to mind is, for one quote from William Osler, medicine is a science of uncertainty and an art of probability. But really, the questions that this case raises are several, and they're relevant for the talk that we are going to take part in today. Can we predict that somebody is at risk of suicide? How do we treat bipolar depression? What is the role of lithium, and how does it compare with other treatments, and are there differences, genetic differences, between responders to these different treatments? And also, we should not forget the question, side effects with long-term treatments. Most patients are at risk of developing side effects. Can we again develop predictive tests that will tell us that somebody is at a high risk of either kidney impairment in case of lithium treatment, or metabolic syndrome in case of, for instance, an atypical antipsychotic medication? Which brings us to lithium. There's a lot of lithium on this slide taken in northern Chile in the Atacama Desert. And what I did, but I obviously did something wrong. But it's been over 64 years since the first modern publication of paper by John Cate from Australia, who first noticed that lithium was effective for people with mania, and was not effective for any other severe mental illness treated in his hospital, where he was the superintendent at the time. And do it manually. And these are some of the people that have been most involved with the history of lithium, especially on the right-hand side, Mogens Skov from Denmark, who has made most of the contribution to the prophylactic story of lithium, and he got the Lasker Award for that in 1988. Now, the prophylactic studies of lithium go back a long time, and this is from the classical paper by Paul Bastrup and Mogens Skov from 1967, showing the trajectories of the illness and response to treatment in a sample of patients followed prospectively. And as you can see, there are some patients where the activity of the illness stops with the treatment, stops completely. And these are people who do recover fully, and if you met them, you wouldn't suspect that they have any psychiatric illness whatsoever. But then there are patients like these two, who in spite of adequate treatment and compliance continue experiencing episodes of depression, mania, rapid cycling. And so the question becomes, how do we differentiate these two groups of patients? So that's one angle that we have looked at, that lithium is not effective the same for everybody, and can we come up with what would predict the response to treatment. The other thing is that bipolar illness, like many other psychiatric disorders, is familial, but it has been a struggle to find specific susceptibility genes. And so the question is, can we actually dissect the genetic heterogeneity of bipolar disorder, identify more homogeneous subgroups of patients that would allow us to better search for susceptibility genes for this condition. This is just a recent update of genome-wide association studies that looked at susceptibility for bipolar disorder. And as you can see, there are some solid and replicated findings that reach genome-wide significance. Typically, the threshold here would be 5 × 10⁻⁸. And the bold names are the ones that have been replicated in more than one study. But also notice that the effect sizes are relatively low. The odds ratios are 2 at best, but typically in the range of 1.05 to 1.2 times. And this is also illustrated on the results from the Wellcome Trust Case Control Consortium data, where number of patients and control subjects have been investigated for susceptibility to various other complex traits. And as you can see, for some of these traits, we are getting very strong signals in several genomic regions, but for bipolar disorder, the results are kind of marginal. By the way, for rheumatoid arthritis, we have very strong signal on chromosome 6. Can you tell what gene that is?
MHC. MHC?
HLA. It's the HLA. Major histocompatibility locus. So typical effect size in these genome-wide association studies is from 5% to 50% increase in the risk for any allelic variant. And also, these are first reports, so there is good chance that at least some of these effects are inflated by the Wiener's curse, by the fact that they become more noticeable in the first reports, and in the replication studies, usually the effects become smaller. And any of these polymorphisms explains very small portion of the overall heritability of bipolar disorder. The heritability has been estimated at 80–85%. So any of these variants does explain less than 1% of the total heritability. And this is illustrated by another example with type 2 diabetes, where there have been now over 50 mapped loci that together contribute to less than 15% of the total genetic variants. So the question is, how do we try to go about this and resolve it? We paid quite a bit of attention to the phenotype definition, and we have done some simulation studies showing how heterogeneity actually reduces the statistical power and the observed effect sizes. We simulated complex traits and looked at what happens if we mix in some non-cases in our case sample, and you can see that the observed effects drop very rapidly, and the sample size correspondingly goes up quite substantially. And we have then looked at this also in the Wellcome Trust Case Control data using example of diabetes. We said, well, if we didn't know anything about the pathophysiological difference between insulin-dependent and insulin-resistant diabetes, and we all mix them together, what would happen if we had a disease with high blood sugar level and no other way of diagnosing or separating these subtypes? And as you can see on this table, when there is no admixture, when we look just at the loci associated with type 2 diabetes, we were able to replicate the well-contrast results. But once you start including more and more type 1 diabetic cases into the mix, the effect essentially disappears, and even the strongest finding disappears with 30% of mixture. So this really tells us that the phenotype definition is quite important for these studies, and if we want to detect a true signal, we have to be careful about who we select for our studies. Move this down. I don't think it's stuck. I don't know whether. So we are in a situation where we are looking at possibly oligogenic or polygenic conditions such as bipolar disorder, and that may be associated with a number of genes, and if we are able to separate these into subtypes associated with fewer genes, perhaps the actual effect size of each of these loci will become larger and easier to detect. So I'll just run quickly through the data that suggests that lithium response does identify a valid subtype of bipolar disorder using the criteria proposed by Robbins and Uze more than 40 years ago. Essentially postulating that a valid diagnostic subtype should be identifiable by clinical features, by long-term stability. It should be separable from other conditions, and then there should be support from genetic or family data and biological markers. The lithium responders, we can go through that very quickly, have essentially a textbook type of clinical presentation. This is what has been described as manic depression in the older literature, with recurrent illness, episodes of mania and depression followed by spontaneous remissions like our patient that we saw at the beginning. And they have often family history of bipolar disorder and good response to lithium. We have completed a prospective study stretching over 20 years showing that indeed if you have good stability of response, low affective morbidity index from the beginning, it stays low throughout the follow-up of 20 years. We were able to show that lithium responders are quite different from lamotrigine responders, especially with respect to their clinical course, where the lamotrigine responders have mostly chronic, non-episodic course, and the lithium responders have all episodic remitting course, with lamotrigine responders having high rates of comorbid conditions in their relatives, while lithium responders don't have much in terms of anxiety or substance abuse. And it seems that it's actually the recurrence itself that may be most familiar. This is from data from a high-risk study that we published a few years ago, showing that actually the parents who had episodic remitting course had mostly children who, when they were affected, had the same. And conversely, parents who had chronic illness course had children with chronic course themselves. And that also correlates with lithium response. And we see that in our recent reanalysis of our clinical data. Again, full responders having fully episodic pretreatment course, and partial responders being somewhere in between, and the poor responders showing really mostly chronic clinical course. I will not go through all these tables. This is only to show that there have been quite a few studies looking at the family history associated with bipolar disorder responsive to lithium, and in most of these studies, the lithium responders have significantly higher rates of bipolar disorder, and also they have higher rates of lithium response among their relatives. This is from a study that looked at response among relatives of our probants, and the blue bars are the relatives, and the empty bars are the comparison patients from a general outpatient clinic. The treatment response is measured on a 0 to 10 scale, but especially number 7 and higher, indicating full complete response. And we see that the blue bars cluster on the right-hand side, and the comparison group patients would have fairly high proportion of non-responders among them. And similar data from our work with Dr. Ian Duffy, again looking at response rates in children of lithium responders, and those parents who had responded well to lithium have children who also responded. The parents who did not do well on lithium have children who also don't do well on lithium but do well on other types of mood stabilizers such as quetiapine or divalproex. And finally, we completed a positron emission tomography study. That was a few years ago before the PET scanner arrived in Halifax, so we sent our patients to Toronto for that. They really liked it, and they underwent scanning. We compared lithium responders, valproate responders, and unaffected relatives of lithium responders, and we found that in one particular area in the anterior cingulate region, the pattern of activation or deactivation is completely different between the responders and valproate responders, and also a similar difference in the dorsolateral prefrontal cortex. So these are data that would suggest and support that the lithium-responsive bipolar disorder is a distinct entity that can be differentiated both from lithium non-responders and from responders to other psychiatric treatments. And in the rest of the talk, I'll just lead you through some of the studies that we have done, starting with candidate gene studies, linkage analysis, GWAS studies, whole exome and genome sequencing that is currently in progress, and some gene expression data. The first, or one of the first analysis that we did was looking at the gene for CAMP response element binding protein, CREB1, which is an important point of convergence of various signaling pathways, and the downstream targets of CREB include number of biologically relevant molecules like BDNF and BCL2. And there was some data from animal work from our colleague Trevor Young that suggests that lithium does regulate CREB. And we also had data from tissue cultures from our patients from lymphoblasts showing that actually the CREB and phosphorylated CREB is upregulated in these lymphoblasts from our patients and their relatives, and doesn't respond to any further stimulation, as if the whole pathway is upregulated. While in controls, we saw that the basal levels were much lower than the patient and relatives groups, and with forskolin stimulation, we get a robust increase in expression. So we tested genes for CREB itself. There are 3 genes, and we found an association with CREB1 gene on chromosome 2. And this gene has been now replicated by several other groups in recurrent depression and in bipolar disorder and in other pharmacogenetic studies. The effect is relatively small, so it is in the vein of the effects that we saw in the GWAS data earlier. Another study that we completed in the lithium responders was a whole genome linkage scan. We included close to 300 subjects from 36 families, characterized by response to lithium, and this is the plot of positive lot scores with some peaks on chromosomes 3, 6, 14, and then also some additional peaks on chromosomes 21 and 17. And with complex traits, it's quite hard to actually proceed with the classical position of cloning approach. So we took a different approach here, and we looked at genes that are differentially expressed in postmortem brains from patients with bipolar disorder, and that map to our linked regions. So for instance, in the 3P region, there are a number of genes that we looked at expression in postmortem brain from 8 subjects with bipolar disorder, 12 controls, and we found that a number of genes were indeed differentially expressed, and then validated the microarray findings with real-time PCR, and some of the genes that map to these areas, chromosomal regions, we then tested in subsequent studies. So the most promising gene that we found was genes for synapsin 2, a gene that is relevant for both neuronal plasticity, synaptic function, brain development, axonal growth, and as you can see, its expression is increased in patients with bipolar disorder, and we saw the same in the real-time PCR data. So that particular gene we looked at more detail in both postmortem brain, and the results are summarized on this slide, showing that perhaps also the gene may be epigenetically regulated, that the histone modification in the promoter area of the gene does correlate with the expression of the gene. Interestingly, in another study, our collaborators in Montreal have found that the gene was also increasing, the expression of the gene was higher in postmortem brains of victims of suicide. And we do have some family data that suggests that the genetic susceptibility to suicide does partially overlap with the genetic susceptibility to bipolar disorder. So these are studies that are based on smaller samples and analyses or investigations of carefully characterized smaller numbers of cases or families. But we wanted to also take advantage of interest in the genetics of lithium, and we have formed consortium, international consortium, to collaborate on genetics of lithium response per se. And we have kind of went the other way, using the brute force approach of genome-wide association studies. We have first analyzed data from over 1,300 cases. 300 came from Halifax here. And now we have collected a replication sample that, together with the first discovery sample, will number over 3,000 cases. So we have about 1,800 subjects in the replication study now. And these are some of the earlier results looking at the genome-wide association. See the QQ plot here for the analysis that took and did not take into account covariates. The analysis without covariates looks more promising. We had to do a lot of stratification and data cleaning because the samples, as you could see, came from different areas, including Japan, and also the Italian samples came from Sardinia. So these are ethnically different subgroups, and so in the end, we analyzed only 1,200 subjects, and these are the first results. The most promising ones would be A gene on chromosome 2, and then another signal is on chromosome 7, which is in an intergenic region. So for now, we just followed the SLC4810 gene, which is essentially a bicarbonate transporter. And interestingly, it's a gene that may have relevance for lithium transport. Lithium is transported across the cell membrane by a variety of mechanisms, and one of the strongest and most important mechanisms is exchange actually for bicarbonate. And although this gene does not reach genome-wide significance, it's approaching 10 to minus 6 significance level only, we feel that it may deserve some more study. Just a few comments on a study in progress. This is whole exome and whole genome in some selected families study funded by Genome Quebec, looking at 500 subjects, 300 probants, and additional 200 family members, looking for rare variants that might be associated with lithium-responsive bipolar disorder. We have been sifting through tons of data, as you can see on this slide. Perhaps for technical comments, the depth of the sequencing coverage is over 100 times on average, and we have been finding, of course, lots of variants as anybody else in this field, and some of them we are following further. So this is, for instance, one family where we sequenced 4 subjects first. These are the blue ovals, the affected family members, and looked at their unaffected relatives using Sanger sequencing. And this is the gene that does co-segregate with the illness in that particular family. It's the serotonin 3A receptor gene that is likely, the mutation is likely quite damaging to the structure of the receptor. And we are actually finding in additional families mutations in the same gene family, the serotonin 3B and 3C receptor genes. So this is one promising lead, but it is only one variant per family, and we cannot see the same effect in additional families. So it will be, of course, a question of how this pans out in the larger samples, and we will be testing this in the Onlygen GWAS data as we go. And finally, I would like to just show you, and that this is something that may approach already clinical application. Study that we have completed with a colleague from Sardinia and with George Robertson here at the Dalhousie, looking at the expression of the spermidine and acetyltransferase gene in lymphoblasts from our patients. There have been a number of reasons why we should look at this gene, because it has been linked to the risk of suicide, and suicide victims have altered expression of this gene in their brains. So we asked ourselves whether actually the gene is regulated by lithium, which is a major anti-suicidal agent. It does reduce the risk of suicide, in our patients 6 to 7-fold. So it certainly makes sense to try to understand whether some of the effect of lithium could be mediated by effect on this particular gene. So we took lymphoblasts from over 60 subjects, and we had cells from people who later, after they had been sampled for their DNA and for their lymphocytes, died by suicide. There are 6 such subjects in Sardinia and 3 in our families here in Canada. These are all, by the way, relatives of our patients that we saw at one point, like the lady at the beginning, and then later we learned that they had died. And Alessio did culture these cells for 1 week, and they were in two groups, cells cultured with the medium and cells cultured with 1 millimole of lithium. And this is what we found. We found that actually in the suicide completers, lithium had no effect on the gene expression whatsoever, while in all the other subjects, either bipolar patients who have family risk of suicide, bipolar patients who don't have family risk of suicide, and healthy controls, lithium does upregulate the expression of the gene, but had no effect whatsoever in the suicide completers. And the effects are actually the same across the Italian and Sardinians, Italian and Canadian samples. We tried to understand whether this had to do anything with sequence variants in the promoter region of the gene. So we had it sequenced in the completers, and in control subjects we have found similar variants or the same variants that you can find in existing databases, but none were associated with the effect of lithium, meaning that it's certainly not the promoter of the gene itself that has to do anything with the effect of lithium, so it may be mediated by other mechanisms. So if we take this together, where are we now? I think the data are quite convincing, and not just our data, but from other groups, that lithium responders do differ from non-responders. They do differ at multiple phenotypic levels, so they're a good group to study from the genetic point of view. And we also know quite a bit about what lithium does, so that's an additional advantage, that we can actually combine the genetic findings with the knowledge of pharmacodynamic effects of lithium, and we can leverage this way the genetic research. At the same time, on a practical level, the individualization or personalization of treatment of bipolar disorder so far still has to rely mainly on clinical signs. The variants that we are finding either have too small effect, so they cannot be practically used, or they have fairly large effect, like we saw with the sequencing study, but the effect doesn't generalize across multiple families. So it's one variant per family, and so we cannot really take advantage of that. We have also started collaborating with a group at the Sanford Bernheim Institute that have been actually able to grow stem cell-derived neurons from lymphoblasts from our patients. So we have a repository of over 1,000 such samples that are available. So far we have neurons available from 12 subjects, 4 responders, 4 non-responders, and 4 controls that are available for more study. Clearly we need still larger samples. We need to extend the existing data collections, and we have we have to take advantage of the new technologies. Look at also brain imaging that seems to also provide some clues as to the susceptibility for bipolar disorder. We have data from high-risk prospective study of children of bipolar parents followed with magnetic resonance imaging, suggesting that there are specific brain regions that may be of particular interest, and we should look at the expression of genes in those particular regions. And finally, I would pose a question to you. Can we actually think of medicine as non-personalized at all? Because any decisions that we make clinically are based on a number of factors. Some of these are implicit, some of these are explicit. But when we are deciding to treat somebody, we are implicitly assuming that without the treatment, the patient will get worse. When we are selecting one treatment over the other, maybe intuitively, maybe rationally, we are choosing between two options, and one of them we consider better than the other. So that is something that we need to keep in mind, and I think as much as we can, we should move from the implicit to more explicit way of thinking about prediction in medicine, expectations about clinical course and selection of treatment. So I'll stop to take any questions, but let me first thank all my colleagues and collaborators, especially the college and also the International Group for Study of Lithium that we collaborate with, and the labs of Girola and Gustavo Turecki that have major role in some of the molecular genetic studies. Thank you.
If you have questions, if you just raise your hand so I can bring the mic over so everyone can hear.
Martin, do you have any animal model? Any animal models with lithium gene knockouts or whatever that look sad or especially happy?
That's a problem, and I think George probably could speak to that as well. How do you tell that a mouse is schizophrenic or depressed? And there have been a number of models that are used for screening, for instance, new molecules for the treatment effects, like the learned helplessness or the forced swim test. But these are more tests of anxiety or trauma and this behavioral despair, essentially. There is one promising model for bipolar disorder that is based on the mutation of the POLG gene, which essentially the mutation then leads to accumulation of multiple mutations in the mitochondrial genes. And the intriguing aspect of this is that, first of all, the mutations can be in a number of different genes, not one single gene. And these mice have significant disruption of their chronobiological phenotype. And it seems that at least in some instances, these disruptions can be corrected by lithium and can be destabilized by antidepressants such as amitriptyline, which is something that we see often clinically, that for instance patients with bipolar disorder, when they are treated with antidepressants, go to develop rapid cycling or switch to mania. But there are a number of other aspects of this model that don't quite fit, but this is probably the best that we have.
Hi, I very much enjoyed your talk, thank you. The B-cell data was really quite striking, I thought. The big difference that you saw between the completers and the rest of the group. Well, clinically, were they distinguishable? Were they all on lithium, first of all? And clinically, did the people that were completers but not responding to lithium show the episodic pattern that you see?
No, these were all lithium treated patients, and clinically we couldn't tell the difference. So this is actually, that's why I thought that this would be probably closer to the real life application than most of our other findings that was shown.
Thank you, also very much enjoyed your talk. For this tragic case that you described at the beginning where the woman was stopped with her lithium because of renal toxicity, have you had an opportunity to look at the pharmacogenomics of these individuals in terms of how they metabolize the lithium, but also what toxicities to predict?
We would like to, and we are collecting the data. Unfortunately for research, fortunately for these patients, the lithium toxicity is not as common. The research is also complicated by the fact that bipolar patients have high risk of conditions such as hypertension, diabetes, metabolic syndrome. So not all cases of lithium or renal toxicity can be attributed to lithium alone. But we are collecting the information as part of the Convigen initiative, and there's probably maybe a few dozen patients with kidney impairment at most in those over 3,000 cases. So it will be a difficult study, but we would like, and some of the genes that have been proposed but not validated yet are in the aquaporin family, but that would be again based more on animal work. That probably is where the animal approach might pan out more than for the actual model of bipolar disorder, but drug toxicity would be something where we would probably do better.
The group were taking the B cells, dedifferentiating them, and then pushing them forward to a normal phenotype. Do you have anything you can tell us about differences that might be seen between the bipolar patients and the so-called controls?
Not yet. We just heard a few weeks ago that these cells have finally grown and are available. If you like them, you know,
yeah, no, that's an interesting approach. The other things that you did mention, this SASYN, the synaptic genes.
The synapsin, yeah. Synapsin too.
Aren't there any mice in which that's been overexpressed?
Yeah, and it's a gene that does, for instance, associate also with epilepsy, especially if the expression is higher. And so we also wonder whether actually we should not look at it in people responding to anticonvulsants. But it had been studied in schizophrenia with very mixed results. In epilepsy, where there is a subtype that is associated with this gene, and we have the data in synapsin 2A with association with bipolar disorder and synapsin 2B with major depression.
So one of the things that I can't quite get my head around is that some of the susceptibility genes are high in families which are at risk for both bipolar disorder and other psychiatric disorders, even schizophrenia. And there's, just trying to think of a couple that would fit into that category, very large proteins, for example, that are involved in, some of them in mitochondrial trafficking as well. So how do you explain that susceptibility genes can be linked to such diverse psychiatric conditions?
Yeah, that's a good question. I think there are probably several reasons for that. One is that we see in the families of our patients a fact called assortative naming. So many our patients have spouses who also suffer from some other psychiatric illness. So it's not uncommon to see families where the illness segregates from both parental sides, and in one case it can be, for instance, bipolar disorder on the father's side and major depression or alcoholism on the mother's side. The other thing is that there is probably a subtype of bipolar disorder that is not lithium responsive, but does overlap with psychotic spectrum disorders. And these are patients who often do better on antipsychotics and atypical antipsychotics. And so in this case, we do often see increased rates of family history of schizophrenia in patients who are otherwise are diagnosed as bipolar. And there are some subtle clinical clues that can actually differentiate. And we do blind assessments of our families. So I believe we are not biased towards diagnosing one more often than the other. But so that's a second. The third is that the many studies that are especially large-scale studies like the GWAS analysis are often based on assessments of patients done just in a cross-sectional manner by trained students, and it's quite unfortunate that somebody spends maybe 1-2 hours with a patient, and then thousands of dollars are invested into biological analysis of subjects who are relatively poorly assessed, and so I think that's partly also a fact. Where, for instance, you can show that some patients in their developmental trajectory may start with an episode of depression. Only later in life they develop either mania or psychotic episodes. So the clinical picture is also changeable, and so it is important, I think, to focus on these more homogeneous subgroups.
Time for maybe 2 more questions. It is 1 o'clock. I know some people have to leave, but this is a great discussion too.
Are there some populations that are particularly susceptible to being non-lithium responders? Like various populations worldwide that may have different response rates that you can compare with?
Not quite. Interestingly, actually, the prevalence of bipolar disorder is about the same in most populations worldwide, and the proportion of lithium responders is about 30%. Again, across the globe, we have comparable data from mostly European countries, but also from Chile, for instance, and it seems like it's quite consistent. So there is not much regional variation.
Thanks, Martin, for a great talk. Really interesting. I was wondering, one of the earliest slides you put up was around the GWAS findings of sort of some of the major markers that have been found so far, the most consistent ones. And you mentioned, I think there was an ANC3, the sodium channel related. Yeah, and then there's a calcium channel related. So I know some people have been talking about this sort of ion channelopathy theory of bipolar that's also seems to be, they wonder about that in epilepsy as well, somebody mentioned earlier. And I was wondering what your thoughts are about that and how that sort of relates or doesn't relate to some of the stuff you guys have found. Because I know in some of the ion channelopathy kind of literature, they do talk about anti-epileptics as being sort of, maybe that's where, the valproate and its action on sodium channels. So just curious what you're thinking about that is, and is that a different group than you're seeing in the lithium group?
Well, no, it's a great question because actually the lithium responsiveness is very episodic. It's an on-off type of presentation, so it makes sense to look at ion channels. Several years ago, probably 7 years ago, we had a project to look at sequence in genes coding for ion channels expressed in the CNS in various episodic conditions, and that included bipolar disorder, essential tremor, epilepsy, migraine, and nothing came in bipolar disorder. So we stopped. And we will go back to our sequencing data. The serotonin 3A receptor gene, that's also ion channel. The CACNA1C and the calcium signaling is actually a very strong candidate, and the whole issue of intracellular calcium trafficking is emerging as very relevant for pathophysiology of bipolar disorder and has links to BCL2 signaling as well.
I think we are going to have to call it quits with that, but thank you very much for that. It was obviously very interesting, and you have a lot of people who are interested in your work, but thanks very much.
Thank you.
I also just want to remind everyone that our next seminar will be on January 22nd with Rob Bico, who will be talking about the human microbiome and the genomics work that he's doing with that. So that will be really interesting. And we have a whole lineup of things from January to June. So next year looks like it will be very interesting for this seminar series. So thank you for coming. Enjoy your holiday. And thank you again to Dr. Alda.
Thank you.